Good morning, everybody. I'm really grateful to the organizers for giving me the chance to present about this system that we've been developing over a number of years. And it's great to see so many friends and colleagues here. Because the talk is short and there's a lot I could tell you about the system, it's important for me to point out that we have a poster in the session this afternoon at lunchtime. And two of the group members, so Guillaume is who's sitting right there, is presenting this poster, and also Sonia Rojic who's part of my labs back there. Hi, Sonia. And she's helps lead the curation efforts. So feel free to approach them and ask them questions, or of course myself. So the motivation for the project I'm going to talk about is that data reuse. I don't think I need to sell that idea to this crowd of all, but just in terms of things that are powered that can be powered by the system are things like contextualizing new results. And so this is a paper that was powered by Gemma from on the left-hand side. We did a few years ago with Jesse Gillis's group. And looking for genes that are differentially expressed in a very sort of generic way. They tend to show up very commonly in studies, and that's something that we were able to uncover by analyzing hundreds of data sets from the Gemma system. And so the idea is that knowing which genes tend to be sort of generic and come up in, are likely to come up in any given study, that maybe you can help draw your attention to other genes that might be a little bit more unique. And another big application would be meta-analysis. So this is an example from a paper we published last year from my PhD student, Alex Morin, who also had a poster yesterday on something a little bit different. But so where we reanalyzed hundreds of transition factor perturbation data sets to look for, to do a meta-analysis and find genes which were repeatedly perturbed by transition factor perturbations. And again, this was powered by Gemma. So I'm not going to talk more about those applications because I just want to tell you about the system. And what we are building on is open data sources that already exist. And so the main one is the gene expression omnibus, and I think hopefully many people here are at least glancingly familiar with it. And it's, again, we are getting most of our data from GEO, and it's great, but if people have actually tried to reuse data from GEO, you often have to do a lot of work yourself to make it ready to use. And I'm not going to go through this list of potential things that you might run into when using GEO or data from GEO, but things like one of the big ones is that the annotations are not standardized. All the data processing is not standardized. There's no additional quality control that's done. They rely on the data submitters to do that, and lots of other complications and corner cases. So the goal for Gemma is to ease the path to data reuse. And this is just a shot of the homepage. The software itself is all available under liberal open source license, as well as the data. And we also had a paper a few years ago when the system was half the size it is now, which gives a lot more detail about the curation and data analysis workflows as they existed a few years ago. Things have changed a little bit. So I refer you to that if you want to get more details. I wanted to move then to talking about what's actually in the system that we've developed before I tell you a bit more about what you can do and also how we built it. So we've decided to focus on 3 mammalian species. We used to support a few more species, but we were sort of stretched for resources, so we drew back on that. When I submitted the abstract to the conference, we had over 18,000 data sets, but since then we crossed the 19,000 barrier, and we're going to pretty soon cross 20,000. So this is a mixture of RNA-seq and microarray data sets. We've been doing this for not quite as long as BOSC has been around, but kind of close. We'll have sort of our 20th anniversary next year. For these species, we have about, last time I crunched the numbers, about a third of the data sets that are in GEO. So it's certainly not everything in GEO, but about 40% of the samples, and you can see the breakdown there for how it goes by species. So just a little bit more about why we don't have all the data sets here and what we're actually looking at. So again, I mentioned that we're focusing on 3 mammalian taxa, and then we've really ended up having a very, very broad but also deep collection of data. So thousands and thousands of different experimental conditions. Just imagine taking not quite random, but data sets from GEO and what you would get. On the right-hand side of the slide, I'm showing just a little peek through the keyhole of our new data browser, just listing, just the top, a few examples of the kind of conditions we have. So of course, again, there's thousands of these that you can access. Because of the interests of my lab and our funding sources, we have prioritized certain types of studies. So we have especially good coverage of the nervous system, but we've also, over the last few years, especially focused on making sure we have small molecule, so drug-like treatment data sets. And also, I mentioned transition factor perturbation, so we've continued to collect those, and there's still more of those in the work. So in terms of things that we don't handle, so a very common problem with microarrays, data sets, which I know very few people would do microarrays now, but a huge amount of the historical data, and also when we started, that was all there was. But very commonly, microarray platforms lack actual information about what the probe sequences were. So actually confirming or getting a confident sense of what genes are being assayed is difficult. Studies that lack experimental design, so there may be just a population sample, are a little bit less suitable for us. There's also data sets that are very small, and things like technology evaluations. So again, like there's some sort of swath of the data that is, I would say, not of interest or eligible for us. So that's one explanation for, the data sets we have tend to be a little bit larger and I'd say higher quality or higher coverage. So single cell data is currently a gap, and I'll talk a little bit more about that later. And I'm really interested in getting people's feedback about our plans to do that. And I always tell people that if there's, looking for a certain type of data that we don't have, please let us know. We have done curation of data sets for people like, oh, I need this data sets on this particular cell type or something. Can you help with that? We have done that in the past. So the curation process, I could give a whole an hour-long talk about this. It's quite complicated, and there's tons and tons of different kinds of issues you run into. So this is just a very high-level overview, and it's the same figures on the poster. On the right-hand side are just some screenshots, which are probably too small to read, but just giving a sense of some of our curation interfaces and then the final output. So we have a pipeline that does as much of this as possible automatically. So downloading the data, entering it into our database based on GEO soft files. We reprocess from raw data for as many data sets as we can. So for Affymetrix microarrays and RNA-seq from raw data. Then we have a bunch of diagnostics and quality control measures that we produce. And so that's what's shown in the upper right, some of the kinds of things we do, and the curators will review that and use that to do things like identify outlier samples, which is very common. So these are data sets that are in GEO, many of them published, that have really glaring outliers. So we remove those. And also batch correction is something we address, and I'll talk about that in a couple slides. Then we do annotation, so I'll talk about that next, and then finally differential expression. So I have a slide on each of those, really, some of those steps I'll try to get through. So first of all, another great resource that we benefit from besides GEO is the bioontologies effort that the community has built up over the years. So we rely on those ontologies for doing our data annotation. Last time I did a count, we have over 15,000 different ontology concepts that we actually use. There's still a fair amount of free text descriptions, which we do try to harmonize. There's gaps in ontologies, and we do submit terms to ontologies for inclusion. That's a very slow process. We have a very small ontology of our own for some specialized concepts, but we're trying not to become ontology developers. One thing I'll just sort of gloss over, but just briefly, that our annotations of individual samples in the system used to be more of a bag of ontology concepts. So starting last year, we migrated to a more RDF-like statement framework. This is the curation interface on the right-hand side, so that this lets us do things like just very simply describe like a drug and the dosage, or the drug and the duration. So this has been like a real benefit to making the annotations easier to use at the other end. I just want to mention again briefly that our search uses basic ontology inference to expand queries. So if you search for brain, you'll get a lot of data sets, but you'll get data sets that don't mention brains explicitly, but they involve the cerebellum or some part of the brain. So, and that's true for all the ontologies that we use. Yeah, so the process of adding these terms to to each sample, and also we add some terms to the data set, that's done by curators. So we train undergraduate co-op students primarily to do that. It's been a really great experience doing it that way. And so the tools really make it very fast and support them choosing ontology terms that are already used and making sure things are harmonized. And we have a very lengthy wiki in-house of our curation guidelines. So next, I'll just briefly mention kind of a complexity that we think is uniquely addressed by Gemma. So is batch information. So addressing technical factors that might obscure biological signals is a real, kind of in all types of data collection, you have to be concerned about, and this is especially true for transcriptomes. But this is almost never recorded. And when I say almost never, like I can think of like a handful of cases we've seen where the batch information is actually included by the data submitter in the GEO record. So maybe that's going to change in the future. I hope so. But we try to infer that from things like date stamps or device information in FASTQ headers. But a lot of platforms, we don't have that. And then we use some heuristics to try to group the samples into what we think are reasonable batches. So whether that truly reflects were the technical batches that were done by by the authors, we don't necessarily know. But we'll use some additional heuristics to decide whether those batches are actually correlated with any strong expression signal in the data. We use principal components analysis to assist with that. So at the end of the day, if data sets end up needing it, we perform batch correction. So the pie charts on the bottom just show that for most data sets, 60%, we do have some kind of indication of what the batches might be from those files, but there's still 40% or so that do not. Of those, you can see there's a large swath that we decide they were done in a single batch, or the batch effects are really small, so we don't need to do any correction, because batch correction is actually kind of a nasty thing to do to data. Very commonly, the batches are confounded with the experimental design. This is like, I forgot to put the numbers here, but what is that, a third of the ones where you have the batch information. So then we can't do batch correction. So that's just in there. Again, like a lot of times when we actually do evaluate batches, it isn't that serious. So we're hoping that, especially if you analyze lots of data sets, that'll come out in the wash. And then there's only about 10% of those, 5% of the overall data or so, where we actually end up performing batch correction. So once we have this data, it's like cleaned, it's assembled and reprocessed, it's annotated, and the last thing we do is a differential expression analysis using the experimental design that we set up. So for this, we have an in-house implementation of the lemma-voom algorithm, which is a very standard method for doing differential expression analysis. So we're not just doing t-tests, we're doing multivariate linear models. So we include continuous covariates in the model when if that's part of the experimental design. So this is something that's again above what I think is available through other resources. And then we store those results at the individual contrast level. So the comparisons of the conditions. And the software supports being able to retrieve this in different ways. And at the right, I have a plot, which is not a very interesting plot in terms of what it's showing, but it's just a bit of a flex of the system where we can, with our R package that we did this, we can do the same thing in the Python package or just with the API to pull out the differential expression results for one gene across like as many studies as you want. And then you can make plots of that data. So this is like a volcano plot kind of thing, but it's not a regular volcano plot where each point would be a gene. All the points here are for the same gene. They're all coming from different condition comparisons. And that's, again, something you can generate in just a few lines of code in a few seconds. Finally, I'll just, we have a whole poster about accessing the data, but I just want to make sure I highlight this again. For people who want to use this, that we have this new browser that we developed that is, sort of inspired by lots of sort of shopping cart kind of paradigms. We don't have an explicit shopping cart, but so you can easily search the data very easily and browse the kind of categories that we put the data sets into based on the annotations. And so you can easily navigate and find data sets that you want. And then that interface actually will generate a little snippet of code for you that you can use in your language of preference to retrieve the data, and then go on to doing more analysis that way. Or you can click on a link and go and find a particular data set and download it just through the web interface. So again, see the poster for more information about that. And so the last thing I'll talk about before summing up is our efforts to introduce single-cell data into the system. So this is something that we've been thinking about for quite a while and planning for, and it's really under active development right now. Thanks. So I really would love to hear people's feedback or input about how they think we should do this. So we want to make this basically fit into Gemma's framework, where we would have studies that have biological replicates and some kind of experimental design. We're going to be using pseudo-bulking at the cell type and subject or sample level for our analysis, so that it basically ends up being kind of like a regular expression data set. We definitely don't want to duplicate efforts of other single-cell repositories like cell by gene. In fact, we want to benefit from those. But the biggest bottleneck we have is cell type annotation, and being able to do that in a sufficiently scalable way. So if you know anything about single cell data, you know that that's often a very big task, is trying to figure out what the cell types are. And there's lots and lots of methods out there for doing this automatically. So we probably have to make some compromises in how we do that to make it scalable, but hopefully also useful. And that's something, again, I'd like to hear people's feedback about. So to sum up, I think Gemma is really a unique resource. I'm not aware of anything of its scale and scope. It hides a lot of the complexity of GEO. We don't have all the data, so that's one downside, but we can add data. We add a lot of value to the data, and it's still under active development. And so in terms of how you can get involved, please use it. You can integrate the software into your own tools and analyses, and we'd love to hear your feedback. So I'll just finish by, again, thanking my lab. So I just won't read the names off there, but again, some people are here at the conference, and I want to acknowledge our funding sources, especially NIH, which has supported this project since its inception, and be glad to take questions.
Thank you. Any questions from the audience? Please.
Thanks for a great resource. I have two questions that will be quick. One, perhaps, do you include genotype? Do you include mice genotypes in the metadata information? And second one, if it's okay, I'm a heavy user of Recount. Why should I switch to Gemma instead of Recount? I would like to hear a bit of
Recount. So I'll answer that. Just actually, about mouse genotypes, you mean like mouse strains and things like that? Yes, we do. Of course, and so for Recount, again, that's only RNA-seq data. So we have a lot of our data is microarrays. I don't know that much about Recount, so I actually should probably know more about it. But I wasn't aware that they do this kind of quality control and also the differential expression analysis and annotation at the level that we do. So I think NIH is also GEO is also reanalyzing RNA-seq data sets, and so that's something that is currently kind of a gap in GEO. So I would give those reasons that that's still something that it doesn't seem to be available elsewhere. Yeah.
Thank you.
Yeah.
Hello.
Oh, hi.
Hi. Thank you for that great talk. I would like to introduce myself. I am Emily Clough, and I am the team lead for GEO.
Oh, hi.
Hi. So it is fantastic to see this work, and I would love to speak with you more about use of GEO. In terms of the batch information, do you have an ideal place you would like to find this in GEO records?
Anywhere? Yeah, so I mean, it'd be really great to have that as one of the sample attributes, just give that a batch one, or it doesn't matter what it is. That'd be where I would maybe natural fit, and as long as it's standardized to some degree, we would be able to use it. That would be really great. There's so many platforms where we just can't infer batches for various reasons. And so even, and of course, like we'd much rather have the data submitter tell us what they think the batches are, because we're kind of making it up.
Okay, thank you.
Thank you. Yeah, I'll talk to you later, I hope. Yeah, we have a great relationship with GEO. We communicate with the team a lot. So Tanya Barrett, we had a lot, who's, I guess, before. So thank you.
Okay.